So this is chapter 11, part 1, and we're going to be talking about the process of meiosis in this chapter. And so before we start, just to give you a little bit of a preface here. So meiosis is the second form of cellular division or cellular reproduction that we will talk about. So again, in chapter 10, we talked about mitosis, and in chapter 11, we're going to talk about meiosis. And so, again, just as a precursor here. So in mitosis, we went from one parent cell to two identical daughter cells, okay? In meiosis, we're going to go from one parent cell to four cells, and these cells are still called daughter cells, but they're not identical. Okay, mitosis, shown over here, will be how our body's somatic cells reproduce. So all of our body cells, except for our gametes. And our gametes are going to be over here. So meiosis is going to be the process that in animals leads to the production of gametes. Okay, so here we go. So up here in this figure, I've got this cute hippopotamus with its baby and this really cute flamingo with its baby. And we've also got other organisms here like plants. And so we know that one of the key characteristics that all living things share is the ability to reproduce. And we've already heard of the two types of reproduction way back in chapter 1. So some organisms will reproduce asexually and some will reproduce sexually. And so just quickly, asexual reproduction produces genetically identical clones of an organism through cell division. And so when we talked about mitosis and we talked about binary fission, the way that prokaryotic organisms reproduce, that's an example of asexual reproduction. There are some eukaryotic organisms that can reproduce asexually. In fact, there are some animals that can reproduce asexually. But for the most part, most multicellular organisms will reproduce sexually, including us, as well as the hippo pictured here and the flamingo. So our focus for this chapter will be on sexual reproduction and how the production of gametes is important in that process. So sexual reproduction is going to be when two individuals of a species come together to form an offspring. Okay, and so in that, the two organisms will each have gametes, which are going to be haploid, which means containing half the genetic information as a somatic cell. So two haploid cells, which we're going to call gametes, or sex cells, egg and sperm, they're going to come together through the process of fertilization to produce the first cell of the next generation, which will be diploid. So again, it has chromosomes from each parent, one complete set from each parent, and so two copies of each chromosome. And that diploid cell will be called the zygote. Now that zygote will then start to divide by mitosis. One cell becomes two, two becomes four, and so on and so forth. Okay, so the rest of the organism's body cells are going to be produced by mitosis. So again, these haploid cells that participate in fertilization, they're going to be produced by meiosis. Okay, these are not somatic cells. Again, they are called gametes. And so again, they're produced by this process called meiosis, which is the focus of this chapter. Sexual reproduction is pretty important in an evolutionary sense. What it does is it introduces variation. And variation in a population is going to be what natural selection acts upon, which is how we get evolution. So the more variation that exists in a population, the better that population or that species chances are for survival. Okay, so we say that the more variation, the higher the evolutionary success of the species. And so again, most eukaryotic multicellular organisms will reproduce sexually. Okay. So some key terms as a reminder, and we talked about a lot of these in chapter 10, but just to make sure we have it. So DNA in an interphase cell, a cell not dividing, will be in the form of chromatin. Okay, so there's that term again, chromatin. What is that? That's DNA, double-stranded DNA, associated with histone proteins. So remember, that's that first level of protein folding of DNA condensation. We saw nucleosomes and linker DNA, if you can remember back to the tail end of chapter 10. When we see chromatin, it looks diffuse, kind of spread out, not coiled. And again, that is how the DNA will appear in an interphase cell. And so right here, I have for you a picture of an interphase cell. So you can see the nucleus is intact. This dark spot is the nucleolus, and the DNA is not shown coiled, instead it's granular. And so again, it's really diffuse and spread out through the cell. During cell division, whether it's mitosis or meiosis, the DNA doesn't look like chromatin. Instead, it fully condenses into chromosomes. Remember, there are proteins involved here, fibrous proteins that help DNA to coil up and condense into chromosomes. Remember, we have monads and dyads. So again, your chromosomes will start off as dyads, two identical sister chromatids joined together at the centromere. And then as we separate them, they become monads, one-armed chromosomes. So if we look back here, this is a dyad with two arms. And if those two arms come apart, they are monads. So single chromatids. And just a reminder of diploid versus haploid. A diploid cell is going to be a cell that has two sets of chromosomes. We represent that by 2n. And those two sets will be homologous. So remind yourself of that term, homologous chromosomes. In a haploid cell, these would be cells having just one set of chromosomes. There would be no homologous pairs. And again, we can represent that by a 1n. When we think about somatic body cells in a human, our body cells are all diploid. The only haploid cells inside of us are our gametes, so egg cells and sperm cells. And so again, the process of meiosis is going to go from a diploid cell to 4 haploid cells being produced. Okay. And so just a reminder of what fertilization is, just so that we have this. Remember again that the fertilization is the union, the fusion of 2 haploid cells from 2 different individual organisms, hopefully not closely related organisms, and you end up with a diploid zygote after that. Okay, so remember, haploid cells contain one set of chromosomes. These would be your gametes, egg and sperm cells. And so here in this picture, we've got a haploid egg and a haploid sperm. Okay, these are our gametes. When these two fuse together, that is called fertilization. The resulting cell, which is pictured here, this is a fertilized egg, will be called a zygote. And that zygote now will be diploid. It will have one set of chromosomes from the egg and one set of chromosomes from the sperm. And so it ends up with sets of chromosomes. Okay, so we would call this cell diploid. Okay. Okay. And I just want to remind you of a karyotype. So again, we saw this back in chapter 10, but just to remind you of what this looks like. So again, most animals and plants are diploid, containing 2 sets of chromosomes in their somatic cells. Somatic cells are all of your body cells, not your gametes. Okay, and when we look at diploid organisms, like what's shown here, this karyotype of this female, I know it's a female because she's got 2 X chromosomes, not an X and a Y like a male would. We see homologous pairs of chromosomes. So remember, that term homologous means pairs of chromosomes. They're going to be the same size, the same length, the same shape. They'll have the same location of the centromere, and they have the same genes. So they have the same genetic loci. They may have the same or different forms of those genes, and those are called alleles. Okay. So here's just an overview of meiosis. And so what's going to happen during meiosis is there's going to be a nuclear division followed by a cellular division. And there's actually going to be 2 rounds of that. In the end, we produce 4 haploid cells, which we're going to call gametes, from one diploid parent that we started with. Meiosis is also known as reduction division, and that's because we reduce the number of chromosomes in the cell by half. So if we're talking about humans, the human parent cell would start off with 46 chromosomes, and after 2 rounds here of meiosis, or two division events, we end up with 4 cells that each have 23 chromosomes. Okay, this reduction in chromosome number is essential to maintaining chromosome number from one generation to the next. Okay, if this didn't happen, then chromosome number could double from one generation to the next, which can't happen. Okay. So before meiosis starts, we have interphase, just like we saw in mitosis. Same stages, G1, S, G2. The most important part here is S phase. And so again, same thing happens, DNA replicates. We go from monad to dyad chromosomes. This happens in that original parent cell. So the same thing that we saw in mitosis happens here. Following interphase, so after all the DNA gets copied, we have meiosis 1, which is the first cellular division. Then we have sometimes this little rest phase called interkinesis, which nothing really happens in. And then that's followed by meiosis 2. Okay, so what is in meiosis 1? So we're going to go through the individual stages, but just as an overview, in meiosis 1, what happens is homologous chromosomes come together. They form something called a tetrad. Then they're separated into separate cells. And so right from the start, we go from 1 diploid parent to 2 haploid cells by the end of meiosis 1. Okay, the phases very nicely are named prophase, metaphase, anaphase, telophase 1. So prophase 1, metaphase 1, anaphase 1, telophase 1. So the same names are used here, which is nice. So by the end of meiosis 1, we've reduced chromosome number by half. Interkinesis, again, is a gap. Depending on the species, it can be very long or it can be non-existent. Okay, so it all depends. That's then followed by meiosis 2. And in meiosis 2 looks just like mitosis. So what happens here is the dyad chromosomes in both of these cells are now going to be separated. And so we go from 2 cells to 4 cells. And each of those 4 cells will have monad chromosomes. And they'll still be haploid. Again, the phases here are prophase, metaphase, anaphase, and telophase 2. So the naming is wonderful. Now for this part of the chapter, our focus is going to be on meiosis 1. And then in the second part of the chapter, we will focus on meiosis 2. Okay, so here's an overview, just taking what was in words on the last slide and putting it in picture form. So again, in interphase, the main thing that's going to happen is we're going to replicate our DNA. And so you can see, where's my pointer? In this first cell, we have 2 monad chromosomes. This is a homologous pair. These will both be replicated. And so by the end of interphase, we still have 2 chromosomes, but they are now dyads. Okay. In meiosis 1, these homologous chromosomes will pair up, and then they'll be separated. So we get 1 dyad of the homologous pair in this cell, and the other in this cell. And then in meiosis 2, the arms, the sister chromatids, come apart. And so again, from this cell we get these 2, and from this cell we get these 2. Okay, so again, our focus is going to be on meiosis 1. Okay, so interphase. Here's a picture of a cell in interphase. Again, it precedes meiosis 1. We've got G1, S, and G2. And again, the most important part here is the S phase. And in S phase, the DNA replicates. So we go from monads to dyads. Something else that happens is the centrosome, shown here, also replicates, just like it did in mitosis. So that's pretty much the same. We then go into prophase 1. Now this is very different. So in prophase 1, the cell is getting ready for the first division. And so some things are the same that we saw in mitosis. So the nuclear envelope breaks down and the meiotic spindle gets assembled at each pole. That's pretty similar. Chromosomes condense and become visible. But here is the difference. So what you can see here, and this is really on the next slide, but I'll talk about it here as well. The chromosomes, dyads, pair up. And so in this particular cell shown here, we have a tetrad, a 4-armed chromosome. There's one, and here's the second. So we say that homologous pairs find each other, and they pair up. And so we go from dyad chromosomes to tetrads. The same thing is shown in this figure down here, except for we just have more DNA. This is a different species. And so here we've got 1, 2, 3 homologous pairs that have all formed tetrads. In humans, we would have 23 because we have 23 pairs. So on the next slide, I just want to show you this picture. So this is one homologous pair of chromosomes that have partnered up or paired up to form a tetrad. Okay, so the dyad chromosomes again find each other, they find their homologue, and they pair up to form a tetrad. This is called, it's called a synaptonomal complex or synapsis. Okay, so homologous pairs find each other and they pair up. They're physically held together by proteins. And in this phase, non-sister chromatids could exchange genetic information. And so that is shown here on this slide. So where they cross over, so again, if this is a paternal chromosome shown in blue and a maternal chromosome shown in red, they are a homologous pair. They will find each other and form a tetrad during prophase 1. Non-sister chromatids can actually cross over. Where they cross is called the chiasmata. And if they cross over, they can recombine. And so you can see a recombination is an exchange of genetic information. That will happen during prophase 1 of meiosis 1 while they're in the tetrad form. This is really important. This is another layer of enhanced genetic variation that can happen in gametes and then, of course, in offspring that inherit those changes. Okay, so a lot happens in prophase 1 of meiosis 1. Tetrads form, crossing over can happen, the nuclear envelope breaks down, and the centrioles move to the poles. During metaphase 1, what's going to happen is the tetrads will line up on the metaphase plate. And so you can see a tetrad again is a 4-armed chromosome. Crossing over can, maybe it had already happened in prophase. They line up on the metaphase plate and they attach to the spindle. Okay, they can line up this way vertically, or they can line up this way horizontally. So this would be 1, 2, 3 homologous pairs. So during metaphase, they line up. Now, how they line up is random. And that brings us to something called independent assortment. And let me move my camera so you can see this. So the orientation of tetrads is random. So if we look here at this figure, so notice here, all of the blue chromosomes, which would be, in this case, maybe paternal for dad, and all of the red chromosomes on the right-hand side of the metaphase plate have lined up together here. That's a possibility. But down here in this part of the figure, this is equally likely. So what's different? We've switched the position of these two chromosomes. They're still lined up as tetrads, right? 4-armed chromosomes. But the orientation of the chromosomes is different. That's going to lead to different results in the gametes. Okay, so if they line up like this, you get 2 gametes over here that have just paternal chromosomes and 2 gametes over here with just maternal chromosomes. But the opposite is true down here. All of the gametes, have a mixture of chromosomes from mom and dad. So that's random. In humans, again, we have 46 chromosomes, which means we have 23 pairs lining up like this. And so if we were to calculate the possible genetic variability due to this random assortment of chromosomes during metaphase 1, we would do 2 to the nth, and n equals the number of homologous pairs, which we have 23. So that means over 8 million possible gametes. So think about that level of variation. Okay, very, very enhanced. So you can get genetic variation from this as well as from crossing over. So there's a lot of enhanced genetic variation that comes with sexual reproduction. This is going to come into play again when we get into genetics. So we'll talk about independent assortment again. Okay, so let's finish this up. Anaphase away. So during anaphase 1 of meiosis 1, the meiotic spindle is going to pull the tetrads apart, and the dyad chromosomes will migrate to the poles of the cell. So again, I've got 2 different representative cells here. So again, these were just together, and now they're being pulled in 2 separate directions. And the same thing is happening here. These were all just together, and now they're being separated. So again, tetrads are separated, and dyads migrate to the poles. During telophase 1, or cytokinesis 1, and cytokinesis 1, I should say, at the end, those separate dyads arrive at the poles. The chromosomes may start to decondense a little bit. The nuclear envelope reforms. And so again, we can see in both, either of these figures, we've got both cells. So we've got a nuclear envelope reforming here and a nuclear envelope forming right here. And so we end up in the end with 2 nuclei. And then cytokinesis is going to give us 2 separate cells. So eventually this will pinch right off. Okay, now both of these cells will then enter meiosis 2, which is in the next set of slides, chapter 11, part 2. So the end result of meiosis 1, we go from 1 cell to 2. The cell that we started with was diploid. The 2 cells that we end with are haploid. There are no more homologous pairs. Each of these daughter cells has just 1 chromosome of each homologous pair. So we've already reduced chromosome number by half. Okay, so again, watch the next video for the second half of meiosis.